Hey there, we are going to be looking at some slides as we go through what to expect for the onion root tip lab. But first, let's take a peek at the anatomy of an onion. So here, this is an onion, and you can see where this is green, and that's going to be where all of our mesophyll tissue is with all of our chloroplasts. And down here, this is where our root tips are, and this is where the slides are going to be from. So right down here, where the roots are going to be growing all the time to try and get more and more water. So it should make a lot of sense that there's lots of mitosis happening there, lots of cell division as the root tips get longer. So we are going to just click out of that. Let's make this bigger. And I want to differentiate between the different stages so that when you sit down to do the lab, you know exactly what you're looking at. So here you can see that there is a difference between this stage and this stage, or let's say this and this. And this looks like a solid nucleus. And really what's happening here is if you think of a ball of thread, when you look at it from a distance, you can't really see a single thread. You can just see the ball of material. And that's sort of what's happening here. So the chromatin is in a relaxed state, and so you can't see any of it individually. But as we move into prophase, you're going to start to see it coiling up, and you can start to see the individual chromosomes appear. And so you're going to see many different stages of this. So you're going to see very beginning stages of prophase, and as you get further, you're going to see it a little bit more and a little bit more. Remember that this is not just separate stages, prophase, metaphase, anaphase, telophase, that they take their time and they go through a prometaphase and a proanaphase, etc., etc. So this is our telophase. And so here you can find the prophase. I'm sorry, this is not prophase, this is the interphase. This is going to be your prophase here as you start to see them individually. And then you can start to see them migrate towards the axis here. Right, this is a good metaphase. As you can see, the chromosomes migrate towards the middle. And then I don't see any anaphase here. This is an anaphase. You can start to see the chromosomes migrate to the side. So you can see those spindle fibers almost sort of pulling those chromosomes as they migrate to the poles. That's happening right here. And then as you go a little bit further along, you can see how these two nuclei are a little bit smaller and a little bit darker, and so they definitely look different from this. This is not the same. So this is a cell that is in telophase, and can't really see a cell plate forming yet, maybe a little bit. And then you can even see a little bit further beyond that as the chromosomes start to relax a little bit here. Let's see if we can check the next slide for some good examples. I'd say that this is a good prophase. Let's see if we can find another metaphase. This is a prophase metaphase, so the prophase is starting to become metaphase, but they haven't totally migrated to the axis yet. So see how there's like it's that prometaphase, and here's an anaphase. This is a good anaphase, and this is another good anaphase telophase, sort of in that between stage. You can still see they're migrating a little bit, but still they are condensed enough that they have sort of separated. It looks like one cell with two separate nuclei here. So hopefully that this has given you an overview of what to expect for the onion root tip lab so that you can correctly identify the types of phases. All right.